Hello everyone, myself Professor Shahana Shengupta from SVIMS. Today I would like to talk on immune system. Let's start with the immunology. Immunology is the branch of bioscience that deals with the study of host defense mechanism. Immunity is the ability of the host to protect itself against foreign organisms, or it also can be called as the state of protection from different sorts of infectious disease. Immune system basically comprises of different types of tissues, cells, along with different sorts of molecules, which altogether mount a kind of response. That response is known as immune response. Whenever there occurred the entry of the foreign invaders from the environment within our body system, then our immune system basically try to mount this kind of immune responses. It is a system that is present in our body responsible for maintaining homeostasis by recognizing harmful from non-harmful organisms and produces an appropriate immune responses. Let's talk about the types of immunity. Immunity may be of two types. Number one, innate, native or natural kind of immunity. And number two, adaptive or acquired immunity. First of all, we will talk on innate immunity. Innate immunity is a set of disease resistance mechanisms that are not specific to a particular kind of pathogen or foreign antigen. That means they are very much non-specific. They are known as the first line of defense of our body system. They are comprises of different types of molecular and cellular mechanism. They are not altered after even repeated exposure from a single kind of antigen or foreign element. Let's concentrate on acquired immunity. Acquired immunity is little bit different from the innate one. It is specific in nature. It is having the capacity of recognition of a specific foreign element, and after recognition it also can eliminate the specific foreign antigen or molecules from our body system. Now onwards, we will talk about the barrier system that is present in innate immunity. Innate immunity can be seen to comprise of four different types of defensive barrier system. They are respectively anatomic barrier, physiologic barrier, phagocytic barrier, and inflammatory barrier. First, we will talk on anatomical barrier system. Anatomical barrier basically includes the skin and the surfaces areas that is connected to the openings of the body system. And these opening parts of the body system mostly guarded with a special kind of mucous membrane. Those are known as mucous epithelial layers. This mucous epithelial layers really acting as a very good anatomical barrier, as a very important tool of the innate immune system. They can be seen in respiratory tract, gastrointestinal tract, and urinogenital tract, along with the ducts of secretory glands. For instance, we can take the example of skin. In skin, there occurs the presence of sebaceous gland, and from the sebaceous gland, there occur a oily secretion known as sebum. That sebum is consisting of a kind of fatty acid, that is the lactic acid, and due to the presence of lactic acid, our skin always maintains a pH range that is between the 3 to 5. This acidic environment inhibits the growth of most of the microorganisms and guard us from different sorts of microbial infections of skin. Hello everyone, myself Professor Shahana Shengupta from SVIMS. Today I would like to talk on immune system. The third important innate defense mechanism is the phagocytic barrier. Now, what is this phagocytic barrier? Phagocytic barrier basically includes the process of phagocytosis. What is phagocytosis? Phagocytosis is a type of endocytosis whereby a cell engulfs a particle to form an internal compartment. That internal compartment is known as phagosome. Within the phagosome, there occurred the further degradation of the particle that is being taken by the phagocytic cell. Those cells are being involved in the process of phagocytosis. They are known as the phagocytic cell. Another set of protein molecule, they are also taking part in the process of phagocytosis. They are collectively known as opsonin. Their main function is to enhance the process of phagocytosis. Now we will discuss about the example of phagocytic cells of the immune system. Many cells are capable of carrying out the process of phagocytosis. Several types of immune cells are particularly specialized in doing this particular kind of functions. Let's talk about the example. First of all, polymorphonuclear leukocytes, or in abbreviation, we can also call them as PMN leukocytes. They are quite efficient in carrying out the process of phagocytosis. Under this PMN, there occurred the presence of different granulocytes, basophils, eosinophils, neutrophils, and mast cells. Rather than these PMN leukocytes, there occur another sets of cells like natural killer cell, macrophages, and dendritic cells. They are also playing an important role in carrying out the process of phagocytosis. Neutrophils are abundant in the blood and important in acute inflammation, as they are the first immune cells to arrive at the site of infection. Another type of phagocytic cells are macrophages. They are sometimes also called as activated macrophages. They are basically resident of the tissues. They are the key as a first defense mechanism and in initiating the adaptive immune response. The third type of phagocytic cells are dendritic cells. They cycle through the bloodstream, tissues, and lymphoid organs, sampling potential pathogens and acting as a major link between the innate and the adaptive immune system. Inflammatory barrier is a defense mechanism of host against shots of infection. It acts against microbial toxins, bacterial infection, as well as mechanical means. It is very helpful in preventing the spread of infection. Basically, inflammatory barriers starts from the tissue damage that is caused by a wound or by an invading pathogenic microorganisms, which induces a complex sequence of events that is collectively known as the inflammatory responses. There occur a sets of events taking place whenever there occur the inflammatory responses. A bacterial infection causes tissue damage, which release a various vasoactive as well as chemotactic factors. These factors induce increased blood flow to the area, increased capillary permeability, and an influx of white blood cells, including phagocytes, lymphocytes, from the blood into the tissues. The serum proteins are also playing a very important role in this inflammatory response mounting by their antibacterial activity. Now we will discuss about the different steps of inflammatory response mounting. The first step is tissue damage and release of histamine. Tissue damage is basically caused by the microbial toxin or by any sorts of mechanical injury, coupled with the release of histamine. The step 2 is vasodilation. Histamine acts on surrounding blood capillaries and causes vasodilation. The third step is increased permeability. At the same time, histamine increased the permeability of blood capillaries, leading to leakage of fluid from blood capillaries. As a result, there occurred the accumulation of the fluid causing edema. The fourth step is known as extravasation. Within few hours, neutrophil migrates to the site of tissue or cell damage by the process of chemotaxis and passes through capillaries wall and enter into tissue space by the process called as extravasation. The step 5 is phagocytosis. With the prevailing condition, now the phagocytes is ready to start with their phagocytosis. Final stage is the mounting of inflammatory responses. As inflammatory response develops, various cytokines and other inflammatory mediators act on endothelium of local blood vessels, including increased expression of cell adhesion molecules or CAMs. The epithelium is then said to be inflamed. Neutrophils are the first cell type to bind to inflamed endothelium and the extravasate into tissues. Hello everyone, myself Professor Shahana Sengupta from SVIMS. Today I would like to talk on immune system. Now we will discuss about the portal exit point of the microorganisms from the body of the host. Respiratory release along with the salivary release, skin kale release, fecal exit along with the secretions from urogenital tract and removal of the blood particles from the affected person all are having the entry of the microorganisms or the infected parts. So they all are regarded as the portal exit point of the microorganisms from the body of the host or the infected person. Now we will discuss about the example of phagocytic cells of the immune system. Many cells are capable of carrying out the process of phagocytosis. Several types of immune cells are particularly specialized in doing this particular kind of functions. Let's talk about the example. First of all, polymorphonuclear leukocytes, or in abbreviation, we can also call them as PMN leukocytes. They are quite efficient in carrying out the process of phagocytosis. Under this PMN, there occurred the presence of different granulocytes, basophils, eosinophils, neutrophils, and mast cells. Rather than these PMN leukocytes, there occur another sets of cells like natural killer cell, macrophages, and dendritic cells. They are also playing an important role in carrying out the process of phagocytosis. Neutrophils are abundant in the blood and important in acute inflammation as they are the first immune cells to arrive at the site of infection. Another type of phagocytic cells are macrophages. They are sometimes also called as activated macrophages. Thanks a lot for your patience. Please do subscribe my channel. Stay tuned for the upcoming videos in different fields of biosciences.